Every single day your body is pulling off this absolute miracle, right? It's keeping trillions of cells in perfect balance. So what happens when just one of those cells decides to, well, break all the rules? Today we are going to dive into the genetic story of cancer, how a totally normal cell can turn into a runaway engine.
And that really is the question at the heart of it all, isn't it? You've got trillions of cells, all working together in perfect harmony, following this incredibly precise set of instructions. So how does one of them just go rogue and start a rebellion?
Okay, so we all have a general idea of what cancer is. It's this uncontrolled growth, right? Cells that just multiply, spread, and invade. But the real story isn't just what they do, it's why they do it. And that why is written right there in the code of our DNA.
So to make this all make sense, let's use an analogy that's going to guide us through this whole thing. I want you to think of each one of your cells as a car. Simple enough. It's got an accelerator to make it go, and it's got brakes to make it stop. For everything to work right, that balance between go and stop is absolutely crucial.
All right, first up, let's talk about that accelerator. In our cells, the go signal comes from a specific class of genes that, when they get messed up, become what we call oncogenes.
Now, in a healthy cell, these genes are called proto-oncogenes, and their job is totally essential. They give the go signal when it's time for the cell to grow and divide. Like when you need to heal a cut. They're the accelerator, but they're used only when needed. It's all perfectly controlled.
The trouble starts with a mutation, a random change in the DNA. And this single little event can transform that well-behaved proto-oncogene into a rogue oncogene. That controlled accelerator pedal, well, it's about to become a huge problem.
This is what's known as a gain-of-function mutation. Think about that. The gene isn't broken, it's supercharged. It's like the accelerator is now welded to the floor, giving the cell this constant, relentless go signal. And here's the really scary part. We have 2 copies of most of our genes, but for this to happen, you only need a mutation in one of them. Just one. So, how does this actually happen? Well, it can be a tiny typo in the DNA sequence that creates a hyperactive protein. Or the cell can just make way too many copies of the gene, kind of like a photocopier gone wild. Or a piece of a chromosome can literally break off and land in the wrong spot, putting the gene into total overdrive. 3 different paths, but they all lead to one very dangerous result: a stuck accelerator.
And believe me, these aren't just theoretical concepts. Famous oncogenes like RAS, MYC, and HER2 are major players in some of the most common cancers we see. Each one, in its own way, is just a different model of a stuck gas pedal, driving that uncontrolled growth in tissues like the colon, the breast, or even in our blood cells.
But a stuck accelerator is only half the story. A car that's in trouble still has another safety system, right? The brakes. In a cell, these are called tumor suppressor genes, or TSGs for short.
These genes are basically the cell's guardians. They are the stop signal. They can pause cell division to repair DNA, and if the damage is just too great, they give the ultimate order for the cell to self-destruct. It's this clean, programmed death called apoptosis. These are the brakes, and they are so important. What's so fascinating is how these brakes fail. It's all explained by something called the 2-hit hypothesis. See, you get one copy of each gene from each of your parents, which creates a natural backup system. So for the brakes to fail completely, you don't just need one bad brake, you need 2.
Okay, so imagine the first mutation, or the first hit, is like someone cutting one of your 2 brake lines. Yeah, this is bad. Definitely not ideal. But the car still has that second line. You can still stomp on the pedal and slow the car down. The cell's backup system is still doing its job.
The second hit, well, that's the catastrophic one. This mutation takes out the second, final copy of the gene. Both brake lines are now cut, and a car with a stuck accelerator and absolutely no brakes, that is a runaway disaster. This is what we call a loss-of-function mutation. The safety system is completely gone. So the cell has a few superstar brake mechanics. The most famous is P53, often called the guardian of the genome, because it senses DNA damage. Then you've got RB, which is like a gatekeeper that decides if a cell is even ready to divide. And you've probably heard of the BRCA genes. They're the master DNA repair crew. When these guardians fail, the consequences are, well, they're severe.
And if you want to know just how vital these brakes are, get this. More than half, 50% of all human cancers involve a mutation in that one single gene, P53.
Just let that sink in for a second. That's how unbelievably critical this one little brake pedal is for all of us.
So, what happens when you have both a stuck accelerator and failed brakes? You get the crash. The cell transforms and it starts gaining this whole set of dangerous new abilities.
This slide just sums it up perfectly. It's the two sides of the same coin. With oncogenes, you gain a dangerous function, and it only takes one bad copy. With tumor suppressors, you lose a protective function, and you have to lose both copies for disaster to strike. One is a stuck accelerator, the other is total brake failure.
So this runaway cell now gets a set of dangerous new superpowers, the hallmarks of cancer. It learns to grow without permission and completely ignores signals to stop. It basically becomes immortal, refusing to die and just replicating endlessly. It even learns how to build its own blood supply and eventually how to invade other parts of the body. It really does transform into a super.
You know, for decades, this was just a terrifying and mysterious process. But now, by understanding the genetics behind it, we're becoming modern mechanics for the cellular engine.
And here is why all of this matters so much. If we know a certain breast cancer is being driven by a faulty HER2 accelerator, we can design a drug to block that specific accelerator. This is the whole revolution of targeted therapy. Instead of just poisoning the entire system, we're becoming mechanics who can actually go in and fix the single broken part.
And it all comes back to our simple analogy, doesn't it? This is the core takeaway. It's a two-part failure. The systems designed to promote growth get locked in the on position, while the safety systems designed to prevent it are completely disabled.
Which leaves us with a final and I think pretty hopeful question. We're getting better and better at reading the genetic story of cancer. So the next great frontier is going to be figuring out if and how we can start to rewrite it.